Today I start my new series of lecture, Physiology. My first topic is cell and membrane physiology. Here we discuss the physiological aspect of cell, not concerned with the molecular characteristics and discovery of cell. Cell. Here the definition is the smallest structural and functional unit of an organism. Now how we can explain the cell or life? A unit which communicate with environment or taking food, excrete waste, divide, increase in size. These all are the properties of cell. Now we talk about structure. Structurally, cell consists a cytoplasm, a fluid containing nucleus, surrounded by a biological membrane. The cytoplasm contain highly organized spatial structure known as organelle which perform essential function for cell including energy metabolism, digestion, protein formation and many more functions. Here in this figure the cytoplasm and nucleus surround with biological membrane. It's the basic structure of cell. In atypical structure, there are many more special organelles present and floating in cytoplasm. A biological membrane, which is also known as plasma membrane, it provides strength, shape, and proper structure to the cell, made up of mainly lipid bilayer and proteins. Other substances which including cholesterol, carbohydrate and conjugated structure like glycoprotein or glycolipid are also present. The lipid which is present in plasma membrane is phospholipid. It contains hydrophilic phosphate head and hydrophobic fatty acid tails. The tails are face each other and form bilayer. Here you see that two fatty acid and another two fatty acid tails facing each other, and here the phosphate head outside and phosphate head inside is present. There are two types of protein present in plasma membrane: the integral protein that embedded in plasma membrane and a peripheral protein which attach electrostatically with the outside or inside the cell. Integral protein provide the channel and act as a transport protein and also act as a receptor, while peripheral protein act as an enzyme, act as an antigen and also sometimes attach with integral protein. Cholesterol, a steroid in nature present in some amount in plasma membrane and that maintain the fluidity. When the temperature is fluctuated, it maintains the melting temperature of membrane or lipids and permeability for some solute. A small water soluble substance may be cross the cell membrane. Cholesterol is present which maintain its permeability or a semi-permeability of the membrane. Glycocalyx. When carbohydrate combine with protein and lipid, it form a glycocalyx. The carbohydrate portion is hanging outside the cell that provide the negative charge that repel different molecules. It also provide attachment with other molecule and other cells, neighboring cells. Transport across the cell membrane. Cell membrane is semi-permeable membrane which selectively allow the molecule to cross the cell membrane. A small molecule which is non-polar in nature like small gases, oxygen, carbon dioxide and other gases are freely cross the cell membrane, while a large molecule which require energy-dependent transport across the cell membrane. There are two types of transport across cell membrane. Number one is passive transport, which doesn't require energy or energy independent transport. Molecule moment of molecule downhill means the molecule is move higher concentration towards lower concentration, while second transport is the active transport which require energy or energy dependent transport, which the molecule is moved from low concentration towards the higher concentration, opposite to passive transport. First we talk about passive transport or energy independent transport, simple diffusion. What is the meaning of diffusion? The diffusion is the spreading of molecule in all direction or the movement of molecule from higher concentration towards the lower concentration. Here we take an example, a glass or a box in which we put a membrane between them and divide it into two parts. Initially the concentration of solutes is equal in both sides. We put some solutes in one side and see after some time we see that it reached the equilibrium. The distribution of solutes is equal in both sides. Now this is the diffusion, movement from higher concentration towards the lower concentration. Another example is that we spray the perfume by one corner of room, and after some time the fumes is spread in all the room. That is the another example that molecules move from higher concentration towards the lower concentration. How we calculate the diffusion? How much diffusion occur in second side when we put some solutes in one side? The diffusion or flow is directly proportional to permeability or area. These two factor increases, the diffusion is increases. When these two factor decreases, the diffusion is also decreases. Here the C1 is the initial concentration or a higher concentration. The second is low concentration is known as C2. We minus initial concentration minus final concentration multiply by area of diffusion and the permeability of membrane. We calculate them and we take the result, then how much diffusion is occur from second side. And minus sign indicate the direction of flow from higher towards the lower. Factor that affect the diffusion. There are many factors that affect the diffusion. The two are the main factor that affect the diffusion. Number one is the area or surface area. Then how much cell or membrane provided area to the solute for transport. Area is directly proportional to diffusion. When the surface area is increases, the rate of diffusion is also increases. When surface area decreases, the rate of diffusion is also decreases. Second one is the permeability. Then how much membrane allow the solute to move across the membrane. It depends on many things like the radius or the size of solute, the membrane thickness. When the thickness of membrane is increases, the permeability is leads to decrease, and the solute size, when the size of solute is less, it is more permeable to cross the cell membrane. Third thing is the polarity. The non-polar substances are freely allowed to move inside the membrane. And fourth one is the partition coefficient, the lipid to water ratio of partition coefficient. When the concentration of lipid is more, because the membrane is a lipid in nature, that allow the lipid to move freely inside. But in facilitated diffusion, the amount of solute is increases, the rate of diffusion is also increases. At certain point it become uniform. Suppose here we have 10 solute molecule and we have 10 carrier protein. Then all the carrier protein is busy to transport 10 all solute molecules. We added another 10 solute molecule. No single carrier protein is free for transport the other solute. That's why at certain point the rate of diffusion is uniform. Number third different is that here in simple diffusion all the substance diffuses freely, but in facilitated diffusion the competition between the molecules occur. Osmosis. Now what is osmosis? Movement of water molecule from less concentrated solution into more concentrated solution across the semi-permeable membrane. In simple diffusion, we put some solvent and we put the solute, we put a semi-permeable membrane between them. We divide into two portions. After some time, we see that the movement of solute molecule from higher concentration towards the lower concentration and reach the equilibrium. Distribution of solute and solvent is equal on both sides. Here the membrane is impermeable to solute, and we see that after some time the water from here is moved to here. Osmotic pressure, the pressure that needs to apply to solution to prevent the inflow of water. Simply, the pressure is applied to prevent the osmosis or the movement of molecule from low ion concentration towards the high ion concentration. Here you see a figure. Here is the pure solvent or water is present. Here a solution is present which contain large amount of solute molecules. The osmosis occur between them. You see after some time the osmosis is going to stop and the solution or the molecule present in solution developed a force, a pressure to prevent more flow of water towards the solution, and the force direction is here towards the solvent to prevent osmosis. You see in this figure osmotic pressure is applied and the osmosis is stopped. Now mathematically the osmotic pressure can be calculated by Ventov law, a law which say that the osmotic pressure is depend of many factor like the concentration or the amount of solute and the concentration of dissociate particle of the solute and the temperature. Osmotic pressure depend upon these three factors. When one of these three factors increases, the osmotic pressure is increases. When one of these three factors decreases, the osmotic pressure is going to decrease. And R is the constant. Our second transport is the active transport, the movement of molecule from lower concentration towards the higher concentration against the electrochemical gradient. So in this process we require energy because we are going to move against the nature of molecule. The nature of molecule from higher concentration towards the lower concentration. We are going against the nature. So we require energy to maintaining it. It's also a carrier mediated transport. In this transport we require a carrier to transport the molecule. Active transport, there are two type of active transport. First one is the primary active transport and second one is the secondary active transport. Primary active transport. Number one is the sodium potassium pump. In this process, the movement of sodium and potassium ions across the cell membrane require energy in the form of ATP. Normally, the sodium concentration is high outside and the potassium concentration is high inside. After some time, the energy stored is more, so the movement of sodium ions from higher concentration towards the lower concentration. Moment of sodium ions from outside to inside and the moment of potassium ions from inside to outside. But we want to maintain the concentration of sodium is greater outside, so we require sodium potassium pump. In one cycle the ATP is bound to carrier protein, and transport 3 sodium from inside to outside and the 2 potassium from outside to inside. Why the 3 sodium is bound? Why not more sodium is bound? Because the portion in carrier protein is 3 available for sodium binding and for 2 potassium binding. That's why 3 sodium and 2 potassium is bound in single cycle, and this cyclic process is continue until the equilibrium is reached. The concentration of sodium is high outside and the concentration of potassium is high inside is reached. Number second, primary active transport is the transport of calcium ions. In intracellular fluid, the concentration of calcium ion is maintained at low level as compared to extracellular fluid, and this is achieved by the transport of calcium ion either in extracellular fluid, either in sarcoplasmic reticulum of muscle cells, or in mitochondria of cells. This is achieved by the pump that present in cell membrane of cell, sarcoplasmic reticulum and mitochondria that maintain the concentration that continuously pump the calcium ion in mitochondria and sarcoplasmic reticulum and outside the cell. When the concentration of calcium is high inside the cell or in intracellular fluid, they bound with different protein and precipitate them. That's why the concentration of calcium ion is maintained at low level in intracellular fluid. Third one, transport is primary active transport of hydrogen. In different body places, the transport of hydrogen is needed. In stomach, the proton pump is present that pump the hydrogen that bind with chlorine and form hydrochloric acid. Second, transport of hydrogen is present in renal tubules that excrete a large amount of hydrogen to maintaining the concentration of hydrogen ion in blood. Another transport is secondary active transport. We discussed previously in primary active transport the movement of sodium outside and potassium inside. 3 sodium move outside and 2 potassium move inside, and maintaining high sodium concentration outside and high potassium concentration inside. When sodium store is large enough outside the cell, they store a large amount of energy, and this energy is higher than the ATP or primary active transport energy. The sodium is diffused across the membrane or downhill to its concentration, and another substance is moved with sodium, and the sodium movement provide energy to another substance or a couple transport of substance with sodium. There are two types of secondary active transport. Number one is co-transport, second one is counter-transport. In co-transport, movement of solute or sodium occur downhill to its concentration and another substance is moved behind sodium or with same direction against the electrochemical gradient. In co-transport, the example is sodium-glucose co-transport present in small intestine and kidney proximal tubules. Sodium transport downhill to its concentration that provide energy for glucose that transport uphill against electrochemical or concentration gradient. And after that, the ionic concentration across the membrane outside or inside is maintained by sodium-potassium pump. Counter-transport or antipode. In this transport, movement of molecule occur in opposite direction. One molecule move and another substance is moved in opposite direction of molecule or solute. It's also known as exchange transport. Here the example is sodium calcium exchanger and sodium hydrogen exchanger. You see in first figure, Here is the sodium move inside and another substance like glucose move with sodium in same direction or behind sodium. It's an example of co-transport or sign port. In second figure, you see here a 3 sodium move inside and in exchange with calcium. Calcium move out of the cell, membrane potentials and action potentials. Our first focus on ion channels and their gating system, opening and closing mechanism of gates. In cell membrane or plasma membrane, integral protein act as a channel, and these channels are selective for transport of different molecule or ions. Its selectivity depends on different factors, which including the shape, size, electrical charge, chemical nature of molecules. There are two type of gating systems. Number one is voltage gating, second one is chemical gating. In voltage gating, the gating control through mechanism change in voltage. When inside cell membrane is more negative, the gate remain closed. When cell membrane loses inside negativity, now gates is open and allow the tremendous quantity of ions to lose outside and go inside. In chemical gating, a binding of chemical occur like a neurotransmitter like acetylcholine or neuro or dopamine bind to certain protein that present in cell membrane or channel that causes conformational changes to bonds and shape that causes the opening and closing of gate and allow molecule to move. In this figure, the gate is present. Now first figure is the voltage example of the voltage gated channels. Here is the ion is present when they receive the change in voltage, they open and they receive the change in voltage, then it is closed. Second example of the chemical gating, a ligand is bind to a protein that causes conformational changes to in bonds and the shapes that causes the molecules to allow to move inside. Electrochemical gradient. It consists of two gradients. Number one is the chemical gradient, second one is the electrical gradient. In this figure, we put some solute here and simply the chemical gradient, movement of solute from higher concentration towards the lower concentration, and solutes are moved from outside to inside. After some time, both side on the solute is in equal quantity and equilibrium is reached, and both side the concentration of solutes are equal. But here we talk about the charged particle. So here is the negative ion is present and the cell membrane is impermeable to negative ion. And the negative ion, when we talk about the charged particle, uh, when there is charged particle is present, so they developed a potential difference. So negative charges attract positive charge and apply the pressure on positive charge, and positive charges trying to move from outside to inside, and all positive charges move from outside to inside. It's override the concentration difference or equilibrium. So inside a large concentration of solutes is present or a charged solute is present. At certain point, a large concentration of charged solute present in inside, and they prevent more movement of molecule from outside towards inside. The electrical gradient, the direction is from outside to inside, and concentration gradient from inside to outside. Both forces are in equal amount and they repel each other. That's why the net moment of solute is zero. Diffusion potential. A potential is generated across the membrane when charged solute move down its concentration gradient. In this figure, we take two solutions and separated by semi-permeable membrane were put in same container. Number one solution contain high concentration of sodium chloride and as compared to number two solution. The membrane is only permeable and only allow the sodium ion to move across the membrane. Then movement of sodium occur from high concentration towards the low concentration. In your next figure, here this figure, we see that the high concentration movement of sodium from high concentration towards the low concentration. And solution 1 loses its positive charge, that's why it increases its negativity. And solution 2 gain positive charge, that's why it's increase net positive charge. So potential that generated during the movement of sodium ion down to its concentration gradient is known as sodium diffusion potential. Equilibrium potential. The potential at which electrochemical equilibrium is established, both forces, chemical gradient and electrical gradient, opposes each other and no net moment of solute. Nernst equation. What is Nernst equation? Where I can use this? Now it's a calculation of voltage or a potential difference. Now which potential difference? A charged particle which is moved from high concentration towards low concentration simply. Now they have certain type of voltage or a potential difference. Now that potential difference is known as the diffusion potential. We discussed previously. Now how much potential difference is more required to reach its final moment or at that point which is no net moment of solute between across a membrane. Now how much potential difference is required? We simply calculate that potential difference by Nernst equation. It's a ratio of concentration difference between two sides, now outside and inside. And this ratio put in log function and multiply by certain things like a temperature multiplied by general gas constant and divide by the number of electrons or charge or ion, like for sodium and potassium charge is 1, for calcium because the divalent ion so the charge is 2, and multiply Faraday constant. In numerator two things, gas constant and temperature. In denominator two more things, Faraday constant, charge. We calculate them and on other side we calculate the ratio and put in log function. After whole multiplication we take a result of potential difference and that potential difference is required for a final moment or a net moment at which point the net moment is 0 or a equilibrium potential. Resting membrane potential. The potential which is about minus 70 millivolts, which means the voltage or a potential difference that present inside the membrane is less than minus 70 as compared to the outside. We see in this figure. First we talk about the solute or a charged particle which is present in outside as well as the inside of membrane. And when charged particle is present, they developed a potential difference. We previously talked about in our previous slide a channels or voltage gated channels as well as the chemical ligand channels. Both are present, voltage sensitive channels and a chemical ligand sensitive channels. We talk about the solute the positively charged particle or some particle which move across the membrane, but some are the permanent particles like protein molecule which is negative and charge, or anion molecule which permanently stay inside the cell and developed a potential, but they can not move across the membrane. And the positive charge particle like the sodium and the potassium is moved across the membrane, and changing leads to change in potential difference. Sodium and potassium pump is also present, which actively pump 3 sodium outside and 2 potassium inside, and maintaining high sodium concentration outside and high potassium concentration inside. With sodium potassium pump, the sodium potassium leak channel is also present. Now, what is the function of leak channel? When certain type of chemical ligand bound with chemical channels or the membrane recept the change in voltage that leads to open of certain type of sodium gated channels and allow sodium inside from high concentration towards low concentration. At that time, a leak channel is present and sodium flow inside and potassium flow outside through this leak channels in tremendous quantity and leads to change in membrane potential, and the resting membrane potential is converted into action potential. Action potential, it's the property of excitable cells, nerve and muscles that consist of stage like a rapid depolarization or upstroke followed by a repolarization of membrane. Initially, the resting membrane potential is maintained at -70 millivolts with high conductance of potassium ions as compared to sodium channels or sodium ions, because sodium channels are closed. The sodium go inside the membrane and membrane loses its negativity, and this process is known as the depolarization. When membrane is going to less negative, it's known as depolarization. As depolarization proceeded, the potassium channel is going to open and allow tremendous quantity of potassium to go outside, because the membrane is want to make its original state or original negativity. When potassium channel is open and allow and potassium ions leaving from the inside, the membrane is going to come back at its original state or original negativity. Hyperpolarization, the potassium conductance remain higher than at rest for some time after the closing of sodium channels, because the inside of cell is want to increase its negativity. That's why we it's need hyperpolarization. It's opposite to depolarization. We see in this figure or a graph, it's the action potential. Here is the stimulus. Stimulus is received by the receptor. The receptor may be present in your eye, or a light receptor which recept a light amount of light, a light stimulus. A receptor is present in your hair which recept the sound wave. A receptor is present in your skin which recept the mechanical pressure or a mechanical stress. Is when receptor stimulus, it's going to depolarization or the generation of action potential, but the stimulus is required in certain amount that cause action potential. When a stimulus is below a certain amount, they cannot cause action potential, and that certain amount, a quantity, is known as the threshold. When the stimulus is below threshold, the neurotransmitter is less released and the channels is less open. They cannot cause the action potential or generation of action potential. After proceeding the depolarization, membrane loses its negativity at certain peak, and that point the potassium channel is going to open and allow tremendous quantity to potassium out from the cell. Refractory periods. There are two types of refractory periods. One is absolute, second one is relative. Absolute refractory periods, it's a period during which another action potential cannot be elicited, no matter how large the stimulus. When stimulus is provided to the receptor and it generates the action potential, at that time, after generating the action potential, the all sodium gates are going to closed. First inactivate and then going to closed. At that time, we provided another stimulus, but it cannot generate a second action potential because the sodium channels are remain closed until repolarization of first action potential is not elicited. Relative refractory period, the period shortly after the firing of nerve fiber. When the absolute refractory period is completed, the repolarization occur, sodium channels are going to settle down. But not all sodium channels is going to set. Some sodium channel require a time for setting. At that time, a second we provided a second stimulus, the action potential action potential is generated, but we require a large amount of stimulus as compared to previous stimulus because not all sodium channels are going to settle down. In this figure, this one is the absolute refractory period. The sodium channels are inactivated and then closed, and after that the repolarization is going to start, and this period is known as the relative refractory period. Here the action potential is generated, but it requires a large amount of stimulus. The cytoplasm, which is present between the nucleus and plasma membrane, it's continuously move around the cell. It's a fluid part which contain minute and large dispersed particle and organelle. The clear fluid portion of cytoplasm, which contain different type of solutes like protein, carbohydrate, lipids, and the sodium, potassium, and different type of minerals, which is known as cytosol, and organelle, which are five main type: endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, and perioxysomes, and many more other cellular structure like cytoskeletons, and many different structure are present in cell. Endoplasmic reticulum, it's a network of tubular and flat vesicular structure in cytoplasm which extend from nuclear membrane to plasma membrane all around the nucleus, connected to nuclear membrane. The tubules and vesicles are interconnected with one another, and its structure, its composition same as the plasma membrane or a lipid bilayer, and also contain large amount of protein. We see in this figure it's a nucleus, it's extend all around the cell, and its two type we will discuss later. Here is the rough endoplasmic reticulum or a RER, and this is the cisternae, the cisterna-like structure of rough endoplasmic reticulum, and it is the tubules, that is the smooth endoplasmic reticulum. The space present here, it's filled with a matrix. A matrix is a fluid which the composition of that fluid is changed with cytosol, different with cytosol. A matrix which is a fluid, and its composition different from cytoplasm, and that matrix contain different enzymes that take part in metabolism of cell. When certain type of substance formed in cell, they transported through the endoplasmic reticulum, where they need. In this way, they also take part role in transport of different chemicals. Granular endoplasmic reticulum. It's made up of membrane which have ribosomes attached to it. We see in this figure it's a granular or a rough appearance, and the rough appearance is by the ribosomes attached to it, which give a rough appearance. And this ribosomes is known as protein factory. The composition ribosomes is the protein and RNA, and they take part in protein formation. After protein is formed, the membrane is take the protein and it's modify. Agranular endoplasmic reticulum. The ribosomes are not attached to it, that's why the appearance are smooth. The tubules are the smooth endoplasmic reticulum, which take part in lipid substance formation and other processes of cell that promoted by interreticular enzymes. Golgi apparatus, it is another organelle present in cytoplasm, a member of transmembrane system. Association with endoplasmic reticulum. Structurally, it is similar like the agranular endoplasmic reticulum, which composed of four or more scattered layer of flat enclosed vesicle. You see in this figure association with the endoplasmic reticulum. It is a structure of Golgi apparatus made up of scattered four or more scattered layer of folding. It is a transfer section of Golgi apparatus. Here is the folding is present. Here is the lumen which contain a matrix, and the function of the Golgi apparatus is when vesicle are transported in Golgi apparatus, it's packaged the substance and processed to form the lysosome, secretory vesicle, and other cytoplasmic components. Mitochondria. The mitochondria is called the powerhouse of the cell because it's make energy, it's extract energy. Without of them, cell unable to extract enough energy from the nutrients, and all cellular function would stop. Structurally, it's composed of a double layer of lipid bilayer and also contain a protein. An outer layer in this figure, it is the outer layer, and this one is the inner layer. The inner layer is invaginate or infold inside, which make the cristae of mitochondria. Another special substance is present which is known as a matrix. It's a fluid which contain the enzyme for energy metabolism. It also contains its own DNA which makes it self-replicative. ATP is formed in mitochondria and transported out of the mitochondria where the need it occur. Lysosomes, these are the vesicular organelle. Formation is occur in Golgi apparatus and then break off from Golgi apparatus and disperse throughout all the cytoplasm. Contain a digestive enzyme which digests the unwanted material or damaged cellular structures, food particle that ingested by the cell, and unwanted matter like bacteria, viruses. And digestion is occurred by the hydrolytic enzyme, which including proteases, lipases, and other enzymes that digest the carbohydrate and other substances. Another organelle peroxisomes, which is structurally similar to lysosomes, but functionally it's little bit different. Lysosomes digest the unuseful substances, but it digests or detoxify the toxic substance that enter inside the cell. Formation of lysosomes from Golgi apparatus, but here the peroxisomes is formed in endoplasmic reticulum. It contains different oxidase enzymes rather than hydrolysis. The hydrolysis enzyme is present in lysosomes. Oxidase enzymes are capable to combining oxygen and hydrogen in harmful substances or remove oxygen and hydrogen from harmful substances like the alcohol and form the hydrogen peroxide. At this point, the hydrogen peroxide is also a dangerous to cell. It's also a toxic. Then its toxicity is decreased by the catalase enzyme. The catalase enzyme is capable to modify or break the hydrogen peroxide into simpler one, the water and oxygen molecule. In this way, the less toxic substance or non-toxic substance is formed from toxic substance. The toxicity is reduced and all our functions are normals. Nucleus, the control center of cell, which control and regulate all its activity with the help of expressing protein. Receptor are present which recept the change and DNA give the response with the help of expressing proteins. So in this way, the protein is formed by the genetic material or the gene or the DNA. Structurally, the nucleus is composed of the two layers, the outer layer and the inner layer. The outer layer is the continuation of the rough endoplasmic reticulum. In this figure, here is the rough endoplasmic reticulum, and the outer layer is the continue with the rough endoplasmic reticulum, and the inner layer is also present that surround the nucleus inside. In nuclear membrane, we see here there are lot of the penetrations or the holes are present. Inside nucleus, the fluid is present which contain the chromatin material or the genetic or hereditary materials. This is the chromatin which arranged randomly. Another structure or a body is present which is known as the nucleolus. The nucleolus is formed by RNA and protein, and also it make the protein and ribosomal RNA. The protein is the subunits of ribosomes. In this way, it's make the ribosomes.